My name is Riley Gauss, and this is my Exam 2 video for Genetics 311. Population genetics is the study of the variation of genotypes and phenotypes in populations over time. One way to examine the genetics of a population over time is using the Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium is the study of the genetics of a population over time when there are no factors that could disturb the gene pool. When in Hardy-Weinberg equilibrium, you assume that the population is infinitely large, there is random mating, all genotypes are equally fit, there is no migration, and there is no mutation. For example, there is a species of fish called the crazy fish, and it lives in Crazy Fish Lake. There are 3 different kinds of crazy fish in Crazy Fish Lake. There are 10 flying fish with the BB allele, 20 camouflage fish with the BR allele, and 15 regular fish with the RR allele, and there are 45 fish total. To verify that my population of fish in Crazy Fish Lake is in Hardy-Weinberg equilibrium, I found the gametic array. The gametic array tells us the frequency of the B and R alleles in the crazy fish population. The next step to verify you are in Hardy-Weinberg equilibrium is to find the genotypic array. The genotypic array is the frequency of each genotype in the population. The final step in making sure your population is in Hardy-Weinberg equilibrium is to find out if the Hardy-Weinberg equation will equal 1 when you put your own numbers into it. Our p-value is the frequency of B alleles, and our q-value is the frequency of R alleles. If we create a Punnett square with our p and q values, it will give us the values to plug into our equation of p squared plus 2pq plus q squared. As you can see, when you plug our values into the equation, they equal 1. This means that it is in Hardy-Weinberg equilibrium. One day, a new predator is introduced into Crazy Fish Lake that eats more regular fish than camouflage fish and more camouflage fish than flying fish. This created directional selection. Directional selection is when a species favors one allele more than others. In Crazy Fish Lake, the more B alleles a crazy fish has, the more likely it is to survive and reproduce. As time goes on, the BB allele will be the only genotype to exist in this environment because it is the most favored genotype to survive. This selection will violate the Hardy-Weinberg equilibrium because it causes a disruption in the gene pool. To find out how the frequencies change after selection, you use a fitness value. Our fitness values in this table are higher when you have more B alleles because the fish with the B alleles are favored. To find the frequency of each kind of fish after selection, you follow the steps in the table shown. You can compare the frequency after selection to the frequency before selection to see how selection affected the population. By looking at the table, you can see an increase in frequency in the BB and BR genotypes because they have more B alleles. The increase in frequency was higher in the BB genotype because it had 2 B alleles instead of just 1. The RR genotype frequency went down because there are no B alleles and less fish survived long enough to reproduce. For this video, I used Dr. Halweg's Week 2 slides, pages 114 through 119.